Good day, this is Glenn Sigulingay, a PhD General Science student. Today I'm going to present a study which is a requirement to the subject Biology of Microorganisms to Professor Dr. Brian Ives Chiaraneta, our professor. This study is the removal and biodegradation of different petroleum hydrocarbons using the Filamentous fungus Aspergillus species RFC-1. This study is made by Adnan B, Al-Hawash, Xiaoyu Zhang, and Fuying Ma. Petroleum hydrocarbons are the most important raw materials for industrial chemicals and energy resources used in daily life. However, they also act as major environmental pollutants. With increasing demand for energy, high amounts of petroleum hydrocarbons from exploitation, refining, storage, and transportation contaminate the environment, especially in the case of large-scale accidental spills. Petroleum hydrocarbons are categorized into four broad classes of chemical compounds. The polycyclic aromatic hydrocarbons are highly toxic, carcinogenic, mutagenic, and teratogenic complex compounds and require several years or decades for recovery. The classes of chemicals that occur naturally in coal, crude oil, and gasoline. They are produced when coal, oil, gas, wood, garbage, and tobacco are burned. Most polycyclic aromatic hydrocarbons, particularly phenanthrene and pyrene, consist of 3 or 4 rings and are medium volatility between gaseous and particle phases in the atmosphere. These molecules can be deposited in surface water and soil where they feature long lifespans. Naphthalene is a ubiquitous environmental pollutant causing diseases. For example, the respiratory system disease, increasing petroleum hydrocarbons contamination has caused significant environmental concern worldwide. The petroleum hydrocarbons are released into the environment and may be subjected to oxidation, volatilization, photolysis, and absorption on sediment and soil particles. Biodegradation is the degradation of the materials into environmentally acceptable products such as water, carbon dioxide, and biomass by the action of naturally available microorganisms under normal environmental conditions. Indigenous microorganisms that can withstand and degrade petroleum hydrocarbons may exist in water, soil, and sediment contaminated by crude oil and polycyclic aromatic hydrocarbons. Therefore, isolation of highly degrading microorganisms from the natural environment is a feasible and significant technique according to Ambrosoli et al. 2005. The abilities to degrade and remove crude oil and polycyclic aromatic hydrocarbons vary with microbial strain and characteristics of pollutants. Some microorganisms can utilize straight, branched, and cyclic alkanes and aromatic hydrocarbons, mainly the low molecular weight polycyclic aromatic hydrocarbons as sole carbon sources. Fungi have certain advantages over bacteria with regard to biodegradation due to their resistance to petroleum hydrocarbons. Filamentous fungi, Aspergillus species, have been received considerable attention as potential candidates for the degradation of a wide range of petroleum hydrocarbons from the environment. Two filamentous fungi isolated from a crude oil-contaminated soil in northeastern Ecuador were efficient for petroleum hydrocarbons biodegradation. Aspergillus and Rhizopus species collected from automobile workshops in Pudukkottai, Tamil Nadu, South India, have a good ability to degrade engine oil according to Sen Muzi et al. 2013. An Aspergillus species isolated from Gulf of Mexico has active capability in degrading crude oil, according to Al-Nasrawi 2012. Aspergillus fumigatus from a contaminated environment shows an exceptional ability to degrade anthracene and can metabolize anthracene effectively. Furthermore, white-ripe fungi Herpes lactus and Pleurotus ostratus can also degrade petroleum hydrocarbons satisfactorily. Except for biodegradation, microorganisms also play a significant role in bioadsorption of pollutants in the environment. Microorganisms usually secrete enzymes to degrade pollutants. Extracellular enzymes from 6 Aspergillus species, which are isolated from crude oil-contaminated soil, efficiently degrade crude oil. Although there are some studies on removal and biodegradation of petroleum hydrocarbons, a few researchers focus on the surface absorption and cell absorption of different petroleum hydrocarbons by microorganisms. Exploration of the fate of organic pollutants in microbial cell and surface bears importance for understanding the degradation mechanism and transport of pollutants. This study on removal and biodegradation of different petroleum hydrocarbons using the filamentous fungus Aspergillus species RFC-1 aimed to evaluate the ability of Aspergillus species RFC-1 isolated from petroleum-contaminated soil for biodegradation and removal of crude oil in 2, 3, and 4 rings polycyclic aromatic hydrocarbons. Also to investigate surface absorption and cell absorption of petroleum hydrocarbons, crude oil, and 3 polycyclic aromatic hydrocarbons by mycelial pellets, cell surface hydrophobicity, biosurfactant production, emulsifiability of mycelial pellets, and to explore intra- and extracellular enzymes secreted by mycelial pellets. For materials and methods. The crude oil from Rumaila oil field was obtained from Southern Oil Company, Basra, Iraq. The surface polluted sludge 1 to 10 centimeter collected from Rumaila oil field, Basra, Iraq also. And purchased from Sinopharm Chemical Reagent Company Limited, Denaturation. And both the phenanthrene and pyrene were purchased from Aladdin Chemistry Company Limited. And for the methods, we have the crude oil was dissolved in trichloromethane, 10 gram per liter. We have the naphthalene, phenanthrene, and pyrene were dissolved in cyclohexane, 2 gram per liter as mother liquor. We have the mineral salt medium, the MSM, contain the following per gram per liter distilled water. We have the 1.0 potassium hydrogen phosphate trihydrate, 0.4 potassium hydrogen phosphate, 0.5 sodium chloride, 0.1 ammonium sulfate, 0.2 sodium nitrate, and 0.025 magnesium sulfate heptahydrate. For plate culture, 15 gram per liter agar was added to liquid MSM. The degradation medium comprised MSM and individual substrates of crude oil, naphthalene, phenanthrene, and pyrene at different concentrations. For strain isolation and screening, surface polluted sludge 1 to 2 centimeter was collected from Rumaila oil field. MSM with 1% crude oil was used to feed the microorganisms with oil degradation abilities in soil samples. 5 grams of soil samples were cultivated in 250 ml Erlenmeyer flasks containing 100 ml MSM supplemented with 1% crude oil as a source of carbon and energy at 30 degrees Celsius, with a shaking at 130 rate per minute for 7 days. Primary inoculums 10 ml were transferred in fresh MSM with 1% crude oil, incubated also for 7 days. The enrichment cultivation was carried out under the same conditions and repeated 4 times totally for 4 weeks. The microorganisms detected in the enrichment culture were transferred onto 15 gram per liter MSM agar plates sprayed with 1% crude oil. Morphologically separated colonies were picked and purified on fresh potato dextrose agar or PDA plates at least 3 times. After that, a 5 mm mycelial disc from the margin of actively growing cultures on PTA plate was transferred into 100 ml of potato dextrose broth or PDB medium in 250 ml Erlenmeyer flask and incubated at 30 degrees Celsius with shaking at 130 rate per minute for 3 days as primary inoculums. Then 10 ml of primary inoculums were inoculated into fresh PDB medium and incubated at the same conditions. After 2 days, mycelial pellets were harvested by centrifugation at 7,000 grams for 5 minutes at 4 degrees Celsius and washed thrice with a sterile MSM. Then the mycelia pellets were resuspended in MSM, transferred to a sterile wiring blender cup and homogenized. For the growth of isolated strains, the optimum conditions were as follows: temperature of 30 degrees Celsius with a pH 7 with 250 milligram per liter crude oil, 50 milligram per liter naphthalene, 20 milligram per liter phenanthrene, and 20 milligram per liter pyrene. To screen oil-degraded strains, crude oil and polycyclic aromatic hydrocarbons, the naphthalene, phenanthrene, and pyrene were used as sole carbon sources. For molecular identification of oil-degraded strain, the isolated fungus was cultured for 3 days in PDB media at 30 degrees Celsius. Mycelia were harvested by centrifugation at 7,000 grams for 5 minutes at 4 degrees Celsius, frozen in liquid nitrogen, and then grouped or ground using a pestle and mortar. Genomic DNA was extracted using the cetyl trimethyl ammonium bromide method described by Doyle in 1993. Internal transcribed spacer or ITS fragments were amplified using 2 primers, the forward primer and the reverse primer. Mixtures of polymerase chain reaction or the PCR reaction contain the following: microliter per 25 microliter, 2 times Chinky Master Mix Blue 12.5, ITS1 F1, ITS4 R1, template DNA1, and DNase, RNase free distilled water to obtain a final volume of 25 microliter. Initial denaturation was performed at 98 degrees Celsius for 2 minutes, followed by 35 cycles of denaturation at 98 degrees Celsius for 10 seconds, annealing at 45 degrees Celsius for 15 seconds, polymerization at 72 degrees Celsius for 15 seconds, and final extension at 72 degrees Celsius for 5 minutes. Thermocycling for PCR amplification was performed according to manufacturer's instruction. PCR products were analyzed using 1.5% agarose gel electrophoresis and purified using oxygen DNA gel extraction kit. Then the obtained purified PCR products were directly sequenced by Chinky using the 3730 XL DNA sequencer. ITS sequences were blasted in National Center for Biotechnology Information. Sequence alignment was performed using the Cluster X or the version 2.0. Phylogenetic tree was constructed using the neighbor joining method, and sequence analysis were conducted using the Mega software, the version 6.0. Biodegradation of crude oil, naphthalene, phenanthrene, and pyrene. A 10% mycelial suspension of strain RFC-1 was inoculated into the degradation medium, 20 ml containing 250 mg per liter crude oil, 50 mg per liter naphthalene, 20 mg per liter phenanthrene, or 20 mg per liter pyrene, respectively, in a 50 ml flask. The flasks were incubated in a shaker at 120 rate per minute and 30 degrees Celsius. To reduce abiotic losses, the flasks were tightly sealed using aluminum foil-lined Teflon screw caps. The control was prepared in the same condition without inoculating mycelial pellets. Crude oil and polycyclic aromatic hydrocarbons extraction and analysis. On the day 1, 5, and 7, triplicate flasks were sacrificed, and fungal mycelia were separated from the medium by centrifugation at 7,000 grams for 5 minutes at 4 degrees Celsius. Crude oil and polycyclic aromatic hydrocarbons in the aqueous supernatant, example those remaining in medium, were extracted with equal volumes, 1:1, of chloroform for crude oil and cyclohexane for polycyclic aromatic hydrocarbons using the liquid-liquid extraction method. The total crude oil was determined spectrophotometrically following the method of Zhao, Wang, Ye, Borthwick, and Me in 2006, using the Alpha 1860 ultraviolet spectrophotometer, Shanghai Lab Spectrum Instruments Company Limited, Shanghai, China, at a wavelength of 245.5 nanometer. The oils and oil products have certain characteristics absorbance at UV range, which the heavy oil corresponds to absorbance wavelength of 250 to 260 nanometer, whereas the clean oil corresponds to the absorbance at 215 to 230 nanometer. For polycyclic aromatic hydrocarbons, the extracted organic layer was pulled, dried using anhydrous sodium sulfate, and evaporated using a vacuum rotary evaporator at room temperature. The extracted samples were finally dissolved in 1 ml of cyclohexane and kept at 4 degrees Celsius for further analysis by gas chromatography. The residual petroleum hydrocarbons in mycelia pellets were extracted according to the method of Shen and Wang in 2011. The mycelia pellets were suspended in 10 ml of Tris HCl with a pH 7.5, 0.01 mole per liter, and ultrasonicated in an ice bath with 3-second pulse and 7-second pause for 6 minutes. This fraction was experimentally defined as crude oil and polycyclic aromatic hydrocarbons adsorbed on mycelia pellet surfaces and absorbed by mycelia pellets. Residual concentrations of petroleum hydrocarbons in the medium and mycelial pellets were determined. For the results and discussion, this shows the time courses of crude oil, naphthalene, phenanthrene, and pyrene adsorbed by live mycelial pellets from 1 minute to 1,440 minutes, trends of surface absorption of crude oil, naphthalene, phenanthrene, and pyrene gradually increased as a whole but occasionally decreased. This result was partially attributed to reversible biodesorption. The highest percentage of surface absorption totaled 43.8% at 180 minutes for crude oil, 50.5% and 59% and 67.2% at 90 minutes for naphthalene, phenanthrene, and pyrene respectively. This showed polycyclic aromatic hydrocarbons were more easily adsorbed by mycelial pellets than crude oil. Phenanthrene and pyrene were also adsorbed more easily by live mycelial pellets than naphthalene. This result was consistent with that of Xu et al. In 2013 using microbial consorption. This result also indicated that the live mycelial pellets adsorbed petroleum hydrocarbons selectively. Polycyclic aromatic hydrocarbons absorption by dead fungal biomass increased rapidly from 1 minute to 10 minutes, whereas the increase in crude oil absorption lasted for 40 minutes. Maximum absorption percentages of naphthalene, phenanthrene, and pyrene by dead fungal biomass were 21.5%, 31.9%, and 38% respectively, and followed the order of pyrene, phenanthrene, naphthalene, and pyrene manifesting the highest hydrophobicity. The results were similar to those of live mycelial absorption, but absorption percentage was lower than that of live mycelia. These findings also indicated the selected absorption of dead fungal mycelia in this process. Absorption of crude oil by dead fungal mycelia gradually increased with time and reached a maximum percentage of 18.2% within 40 minutes. The absorption of crude oil in the three polycyclic aromatic hydrocarbons increased with pH values ranging from 3 to 7 and decreased at pH 8. The highest absorption of petroleum hydrocarbons by mycelial pellets of the strain RFC-1 was occurred at pH 6 to 7 and significantly decreased at pH 8. As you can see in figure 3a, this finding suggests that increasing hydrogens inhibit the absorption of petroleum hydrocarbons to cation binding sites. The lower petroleum hydrocarbons absorption at acid pH may be due to the competition for cation binding sites between hydrogen and hydronium ions in the solution or reduced solubility of petroleum hydrocarbons and or precipitation of ions. The effect of temperature 10 to 40 degrees Celsius on absorption of crude oil, naphthalene, phenanthrene, and pyrene by the strain RFC-1 was determined at pH 7, as you can see in figure 3b. Absorption increased with temperature ranging from 10 to 40 degrees Celsius, and optimal temperature for absorption was 30 degrees Celsius. The increase in absorption with specific temperature range was possibly due to the increased energy efficiency in the system, facilitating the attachment of substrates on the cell surface, whereas the decline with a further rise in temperature possibly resulted from the distortion of some cell sites available for substrate surface absorption or adsorption that may occur with high temperatures. Mycelia pellets exhibited a remarkable capacity for absorbing crude oil, naphthalene, phenanthrene, and pyrene. After 60 minutes, absorption capacity increased with time, as you can see in figure 4. The highest absorption percentages of crude oil, naphthalene, phenanthrene, and pyrene by mycelia pellets amounted to 35.7%, 67.7%, 83.9%, and 88.9% at 1440 minutes, respectively. These values were higher than their surface absorption percentages. Absorption and surface adsorption of naphthalene by mycelia pellets were lower than those of phenanthrene and pyrene. This absorption process may be associated with biodegradation. Hydrophobicities of mycelia pellets were cultivated in the crude oil, naphthalene, phenanthrene, and pyrene medium reached more than 70% on the first day and 54% at 7 days, implying the notable emulsifying efficiency and high degradation activity of mycelia pellets. Some researchers have reported the relationship between cell surface hydrophobicity and degradation of crude oil by microorganisms. Hydrophobicity of mycelia pellets reduced gradually during degradation. This decrease was probably attributed to the gradual depletion of the carbon source caused by substrate degradation by mycelia pellets, thus weakening the emulsifiability of mycelia pellets for petroleum hydrocarbons. In addition, hydrophobicity may be associated with substrate concentration and toxicity. However, the decrease in hydrophobicity is probably related with the production of biosurfactant, which can alter the hydrophobicity of the surface and in turn affect the adhesion of microbes over the surface. Petroleum hydrocarbons removal includes not only biodegradation but also the transport and accumulation. In this study, the amounts of petroleum hydrocarbons removal, biodegradation, and bioaccumulation adsorbed onto cell surfaces and absorbed into cells were examined individually to illustrate the relationships with the biological processes. The amount of petroleum hydrocarbons remained in the degradation of medium inoculated with mycelial suspension decreased substantially on day 1, as you can see in figure 6a. Phenanthrene and pyrene decreased rapidly, but crude oil and nap decreased slowly, especially after 1 day exposure. To disclose the degradation mechanism of petroleum hydrocarbons by strain RFC-1, intra and extracellular enzymes were extracted and used for treatment of crude oil, naphthalene, phenanthrene, and pyrene. Degradation efficiency of crude oil, naphthalene, phenanthrene, and pyrene by intra and extracellular enzyme solutions followed the order of NAP to crude oil, pyrene, phenanthrene, and increased over time in varying degrees. The diverse parts of the enzyme perform differently for the different substrates. From days 1 to 7, degradation efficiencies of crude oil, naphthalene, phenanthrene, and pyrene by the extracellular enzymes were slightly higher than those by intracellular enzymes. And for the conclusions, Aspergillus species RFC-1 can absorb and degrade crude oil in the 3 types of polycyclic aromatic hydrocarbons: surface adsorption and cell absorption of crude oil, naphthalene, phenanthrene, and pyrene reached 43.9% and 35.7%, 50.5% and 67.7%, 79% and 83.9%, and 67.2% and 88.9% respectively, and follow the order crude oil, naphthalene, phenanthrene, and pyrene. Adsorption content by heat killed mycelia pellets was constant after 40 minutes of crude oil and 10 minutes for polycyclic aromatic hydrocarbons. Constant adsorption percentages of crude oil, naphthalene, phenanthrene, and pyrene by heat killed mycelia pellets amounted to 18.3%, 21.1%, 31.2%, and 38.3% respectively. Biodegradation efficiencies of crude oil, naphthalene, phenanthrene, and pyrene totaled 51.8%, 84.6%, 51.3%, and 55.1% respectively within 7 days of incubation. Rapid accumulation, adsorption, and absorption, and degradation of petroleum hydrocarbons by the strain RFC-1 indicated its potential application in bioremediation of contaminated environments, including oil-contaminated conditions. The intra- and extracellular enzymes of the strain RFC-1 can effectively metabolize petroleum hydrocarbons, whereas extracellular enzymes play a more important role. This study can provide theoretical support for bioremediation of petroleum hydrocarbons pollutants. And thank you for listening.